Okay, so we're gonna pick up where we left off before with the cancer lecture. So we had basically gotten through oncogenes, which are one of the major ways of making cells cancerous, and now we're talking about tumor suppressor genes, which is the other molecular mechanism. All right, so the idea for behind tumor suppressor genes came from cell fusion studies. So if you take a normal cell, it's not cancerous, and you take a tumor cell, and then you fuse them together, then you end up with a hybrid, but that cell is not cancerous generally. So, which is surprising because oncogenes act dominantly. So you would think that the tumor cell probably has multiple oncogenes activated, and then when you mix it with a normal cell, well, it should produce a cancer. But in fact, they're generally not cancerous. So that tells you that there has to be something that's acting recessively in the tumor cell that, like if two things were knocked out in the tumor cell, well, there's going to be wild type copies in the normal cell that are preventing the transformation or the becoming cancerous. So those things that are acting recessively were called tumor suppressor genes because they're suppressing the tumor. Okay, so the idea is that in a cancer, the tumor suppressor genes are knocked out, so they're not suppressing the cancer. But in a normal cell, they're active and they can suppress the cancer when they're fused with a cancer cell. Okay, so we'd already gone over RB as part of the G1 to S phase transition, right? It is binding E2F and its phosphorylation by the G1 CDK cyclin release E2F, and then you get into S phase, you start making S phase genes. So, okay, but it turned out that in fact it was the first tumor suppressor gene to be cloned, and so basically it's a form of retinoblastoma. So I don't have a picture here of the retina. I had it before, but it's basically the back of the eye where the the retinal cells are, the rods and the cones that are detecting black and white or colored light. And so it's in there that you get the retinoblastoma. And so 1 in 20,000 children have retinoblastoma where they have these mutations in the retina. And there's the thing about retinoblastoma is it's sort of it's like a tissue where you get a solid tumor that you only need one hit. Okay, you only need to hit retinoblastoma and you'll get it. Whereas most tissues, if you inactivate retinoblastoma, nothing happens because it's not sufficient to do anything. But in the retina, one hit and you'll get an retinoblastoma, the cancer. So it has a hereditary form, which tumors in both eyes. It has spontaneous form, and this sort of weird mix of the inheritance of this led Alfred Knudsen to the two-hit hypothesis. So this is so this is one of the few examples where getting some data, really sketchy data, and he could come up with a hypothesis in biology. So normally, biology's you can't really predict what's going to happen based on, you're basically just looking at, maybe he's doing that too. But like in physics, like Einstein, he comes up with his special relativity, general relativity, he's just thinking about it, first principles, right? And he can map it out, oh, it's going to work this way. But in biology, you never know how evolution is actually going to do it. So you could say, oh, this is the way I would do it if I was a cell. That would be like the best way to do it. But in fact, it's not the way that evolution chose to do it. So you can't really do it. But anyway, this is one of those rare examples where this paper was published years before they had actually cloned the tumor suppressor gene. So it's the two-hit hypothesis. And it's based on observations. So here we have a man and a woman. Let's say the man has retinoblastoma and they have children. They have 3 daughters and a son, and 2 of the daughters will end up with retinoblastoma. So this is a familial case, and in a familial case, you're going to end up with, like this is the back, we're looking at the retina now. You'll end up with lots of tumors, and you'll have that in both eyes. Okay? Whereas spontaneous, there's no familial history. You would only have one tumor in one eye. You wouldn't have more than one, and that would be a rare event. But if you have this in your family, then you're going to have multiple hits in both eyes, guaranteed. Okay, so what this tells you is it looks like it's acting like a dominant because half the children get it, right? So it looks like it's acting dominant, and yet if it was acting dominant, why would you have these tumors in only a few cells in the retina? Well, that doesn't make any sense. Every cell, if it's acting dominantly, like let's say it's an oncogene type thing, right? Well, every cell in the retina is going to get it. So why would you only have a few tumors? Now this is more than one, right? It's not spontaneous, but it's still not that many, right? So this is his idea. His idea was, well, sure, it looks like it's acting like a dominant, okay, but molecularly it's a null. So what he's saying is you've got RB minus over plus. You can just say RB plus like that, right? So this is what this person is. Okay? So almost all the cells are going to have one good copy of RB and a mutant copy. And what he's saying is that if this transitions to RB minus over RB minus, and again, what are these things, right? Like, what are those anyway? Like, why are there two of them? Huh? Yeah, but where do they come from? Say it again? Yeah, so where did the diploid come from? Yeah, it comes from each parent, right? So the mother may have given the RB minus and the father gave the RB plus. So you're always going to end up with two copies, right? So you just draw it like this in a genetic diagram. So basically, in some cells, you're going to knock out just spontaneously, not spontaneously, I mean, it is spontaneous, but there's certain mechanisms we're going to go over. You're going to knock out the plus copy. And then you end up with minus. And that's what these tumors are, okay? That's why the tumors don't happen in every cell, because they're just, a rare event where the other one gets knocked out, but yet the whole thing acts like a dominant, okay? So it's acting like a dominant, but it's really a molecular recessive. Okay, so because this, you can follow this in families. Certain families would always had a deletion of chromosome band 13, so it's chromosome 13 band, so there's two arms, Q and P. And it was arm Q and band 14. So they would notice that, in certain families, there was this deletion based on that karyotype where they put out the mitotic chromosomes, where that band was missing. And these are just Giemsa bands. They're just staining, and the bands turn out dark. So based on getting deletions coming in from this side and this side on the family, they were able to map it finer and finer because, basically, these people had retinoblastoma. And then this is a deletion. And this one has retinoblastoma. And then this one has a deletion like this. It has retinoblastoma. And this one has a deletion looks like this, retinoblastoma. So then you say, well, what is the part that's mutated in all of them? And so they just positionally cloned it based on that. And there were two groups. And they had a race and one with that. Tell me a question. Oh, sorry. I'm just saying that these deletions are knocking. So usually you would draw a deletion as not being there, but the line is the deletion. And so that would be like the top one would be in one family, have that type of deletion. The other one had this deletion. So we're just mapping where the deletion endpoints were. And if you have enough deletions, it'll end up limiting the location of the RB gene. So like the RB gene would be right there. I mean, they didn't know what it was. I mean, they called it the RB gene, but they didn't know what it was molecularly. They didn't know what the sequence was. So when they got it small enough in the small region, there's only like one or two genes there and they can test them. All right. So this again is that positive. This is showing you the dominant inheritance. And then this is what I just drew out where in the hereditary form, you're going to have a much higher likelihood of knocking out the second copy because you're starting with one bad copy. And in the non-hereditary form, you're much less likely. I mean, here you have to knock out two copies. So it's much more unlikely because here you already started one copy is mutated. So it's relatively easy to knock out the other one. Or at least you can see how many times it happens, right? I mean, you can just count how many times it happens by, counting how many retinoblastomas there are generally in patients, right? Okay, and this is showing you that the band is getting deleted in these retinoblastoma patients. Okay, so what we're going to talk about now is something general. It's not linked to the RB gene anymore. What we're going to talk about are what are the ways, if you have one copy mutated, to mutate the other copy. So again, we're talking about chromosome 1 from the father, chromosome 1 from the mother, or in this case, it's chromosome 13. So one chromosome 13 from the mother, one chromosome 13 from the father. How do you knock out the other copy? Okay, so in this diagram, the green is mutated and the red is normal. Okay, so there are different ways to mutate the other copy, and it turns out that having one copy mutated will make it more likely to knock out the other copy. Okay, and we can look at that. So one of the ways is non-disjunction is going to cause chromosome loss. We already went over this when we were talking about mitosis. So non-disjunction occurs when, let's say, the mitotic spindle checkpoint's not working properly, and two copies, in this case chromosome 13, go to one cell, and then and no copy go to the other one. But remember, what are these? Okay, they're not the father and the mother chromosome. Okay, that's meiosis. This is mitosis. So this is the chromosome 13 from the father with the copy of chromosome 13 from the father from made during S phase. So when this one goes to this one, this other cell still has chromosome 13, but now it's from the mother, right? It just lost the what's called the sister homologue. So it only has one chromosome 13, not two. Okay. And so then if you, so here, what they're saying is that losing that one chromosome 13, that cell's probably going to, I mean, we're not talking about RB in particular, okay? We're just talking about in general for tumor suppressor genes. The ones that lose that is probably going to die. But this one ended up with extra copy. This is another way. So this may or may not happen. Depends on the size of the chromosome, right? So and then here, what they're saying is that if you've got a mutated copy, and you've got a wild type copy, you end up with two wild type copies and one mutant copy. So we're not dealing with the left side, we're dealing with the right side. So it's got extra chromosome 13. So it turns out that a cell with 3 copies of chromosome 13 is probably going to lose one of the copies just spontaneously. Now, it could lose the bad copy, right? Or, I'm sorry. Okay, so let's step back. Okay, so in this one, the mutant is the yellow. So now you end up with 2 mutant copies and a good copy because you had this the mutant copy was the one that went to this cell on the right. So then you're going to lose, the cell's going to lose one of the chromosomes, so it just has 2 copies, and it could lose one of the mutant copies, in which case it'll go back to where it was before. So it still has 1 good copy in the blue and 1 mutant copy. But by chance, the cell might lose the good copy, the wild type blue copy. Now it ends up with 2 copies, but they're both mutant. Okay, so in this way, you can see that having 1 bad copy made it more likely to knock it out. Okay, so this is the non-disjunction causing chromosome loss, but then you could also have this one is sort of the second one that we talked about here, where you ended up with this non-disjunction and you ended up with that. You're not going to really be able to duplicate that whole chromosome. So despite what these words are talking about, we're really talking about that scenario here. Okay, you can have mitotic recombination event. So it turns out that this homologous recombination across chromosomes So it's basically just happening in meiosis unless there's DNA damage. And then you can have this kind of crossover, and then depending on how they're segregating, you could end up segregating, because the bad copy to the cell that already is getting the bad, already has the bad copy, and now you lost the good copy. Okay, so you can work this out for yourself, but it's basically they're swapping the arms, and now you have a good copy with a bad copy, and if you end up putting the bad copy to the cell that's going to get the bad copy, now you have two bad copies. On the other side, you're going to end up with a good copy and the other situation. So all right, do I need to map that out or not? Yes. All right. Okay. All right. So here we've got a situation where. Oh, sorry. All right. So let's say here we got a mutant situation, and then you have the wild type. Okay, so and now you're going to get a swap. So the wild type is dark. All right, and now, so let's say some DNA damage here where it breaks it, and it's going to end up using this one to replace it. So now you're going to end up with a situation where you have okay, so you're going to end up with a situation where there's a recombination that's occurring within the cell, and then you're going to end up, you've got a situation where you've also got these chromosomes are now going to line up in mitosis. And let's say that you're sorry. Okay. So they're going to line up in mitosis, and let's see how to draw this. We're going to end up, so one, we're going to be segregating the wild type. So you've got another copy of this thing, okay, and it's here. Okay, and then this is let's say this is from the mother, and this one's from the father. All right, so this one's wild type, and this one's mutant, and this one's mutant, and this one's wild type. So now they're going to pair up based on which chromosome they are. Like chromosome 1 from the mother or father are going to pair up together, and then from the father are going to pair up together. And then if you put these two in one cell, you end up with a situation where that one's mutated, and on the other chromosome, it's mutated too. And then over here, you're going to end up with a situation where you've got two good copies. Okay. So basically, they're going to segregate based on being sister chromatids to each other. But this recombination, which had to have happened, so this had to have happened after replication. Oh, sorry. So this had to have happened after replication. So you already had two copies of each of them. And then you end up with like that. So you got recombination among one of the copies, but the other one is not mutated. But these two are going to pair together here. And then these two are going to pair together here. And then you're going to end up, just by chance, getting mutant and mutant. You could flip these, and then you go back to where you were before, just with a recombination event. So, but, if this rare event occurs, well, it's going to turn into cancer. So the thing is, like, it sort of selects for itself because it'll happen. Whereas if it had been flipped, it wasn't going to happen. You still end up with a wild type of mutant situation, which is fine because one copy of the tumor suppressor gene is enough to block the tumor. Okay, so gene conversion is pretty similar. I'm not teaching recombination section of this class, but basically what happens is if you get a deletion on one chromosome, the other chromosome is going to be used to fix it. And there's two ways to resolve that. It's sort of complicated, but you can, but depending on which strand they're cutting, they're going to be either putting back, they're actually going to be swapping the whole chromosome, like going from this one over to this strand, or they're going to go like, they're going to go like this, and then they're going to come back to this one, or like in this case, all right, it goes like this over here and then back to here. So it just really depends. I mean, it's a little bit complicated. We don't need to go into it, but it's basically another way of resolving the mitotic recombination, so that instead of picking up the whole other chromosome like this happened, you just pick up this one section and then you go back to the same chromosome. But it's too complicated to go over now. But it's called gene conversion. But it's basically caused by recombination also. And then if you have this one mutated and you just get a deletion here, then you're going to knock out the other gene. And then of course if you get a point mutation and this one's already deleted, then you're going to lose both copies also. So these are the different ways, and you can see that the deletion is sort of an independent way. This thing is just being deleted independently of what's on this other chromosome. The same thing with the point mutation. But in terms of these ones, you actually have to have the other one mutated to get the mutation on this one, okay? Because in fact, it's the same mutation. The mutation from the mutated chromosome is actually now on both chromosomes, okay? So that's why it's much easier to get, if you have one mutation, these other mechanisms only work if the other copy is mutated to begin with. This one would still knock out this one because it's an independent event, and deletion or point mutation would just knock it out. I mean, sure, you still wouldn't have retinoblastoma unless it were knocked out to begin with, but it's an independent. It has nothing to do with the presence or the type of mutation on the other chromosome. So because of these mechanisms, especially these four, once, this isn't just the fact that these familial forms have so many more mutations, and you compare it to spontaneous, which there's no hereditary link, and they just have one mutation. And here they had to go from RB plus, RB plus, to RB minus, RB plus, to RB minus over RB minus. So they have to have two totally independent events. But here, because these things, so the chance of this happening is pretty remote, because you have to have this random event to make a deletion, point mutation, or something in this one, and then you have to knock out the other one too. Whereas here, because once you have one bad copy, it's easier to, a lot of these mechanisms just transfer that bad copy to the other chromosome. And so you're going to have a much higher rate of knocking out both copies when you start off with one bad. Okay. So there's other ways to knock out tumor suppressor genes, and one of these is epigenetic silencing. So in epigenetic silencing, you can have DNA methylation on these CpG islands, which you guys probably learned about. And so the epigenetic, so you can have one chromosome mutated, but if you condense the other allele into heterochromatin, it's not going to be expressed. And so that's also going to knock it out, okay? And so, here we're looking at, mutations in red or epigenetic changes. And it doesn't matter whether you have two genetic changes or two epigenetic changes or a mixture. you still have knocked out both copies. And the percentage of this epigenetic mutation of different alleles. So here we have different genes that are tumor suppressor genes. BRCA1 is the famous tumor suppressor that's knocked out in breast cancer. And because what you see is like in breast cancer, well, it gets knocked out. But in colorectal cancer, it doesn't matter whether you knock out BRCA1 or not. Actually, that's actually the case. That's why it's negative. But this is the percentage of methylation of these alleles. So it can be pretty high. I mean, well, it's like 15%. So most of them are going to have to be mutated, but in this case, but sometimes it goes up to as high as 40%, depending on the allele. And then, whether or not that allele gets mutated in that particular cancer is sort of the thing. So like like BRCA1 really doesn't have any effect on colorectal cancer or stomach cancer. So it's not really, even affected. But basically, what this tells you is that a sizable percentage, like 5 to 50, 40% or something like that, these tumor suppressor genes can be knocked out just by their conversion through epigenetics to heterochromatin, generally through methylation. Okay, and this is just to remind you what the RB gene is. Again, it's inhibiting the E2F transcription factor that's required to turn on S-phase genes once it's released. It gets multiply phosphorylated by the G1 cyclin CDK complex, makes it release E2F, turn on S-phase genes. So it's regulating the cell cycle, because whether the cell decides to go into another round of the cell cycle or not is made in G1. And so if RB is not there, it's much more likely for the cell to just keep dividing if it's already dividing. Okay, so then the other major tumor suppressor gene is P53. We already saw P53 when we were talking about the DNA repair checkpoint. So basically, if there's DNA damage, there's a replication fork, and we already talked about how P53 is constantly being degraded by the MDM2 ubiquitin ligase, but then the MDM2 ubiquitin ligase is shut off, or at least it can't work because P53 is actually phosphorylated, which blocks it from being degraded. And then now you accumulate enough of it, it becomes a tetramer. P53 can only work as a tetramer, so it has to have a high concentration enough to become a tetramer. It produces P21, which is CDK inhibitor, CKI, shuts down CDK. And we already talked about this pathway not being physiologically relevant for shutting down DNA replication. So this is just a rehash of what we already went over when we were talking about cell cycle. But the main thing we're going to be focusing on here is that P53 is mutated in 80% of human solid tumors. So generally, on your way to becoming a malignant tumor as a solid tumor, P53 is going to be knocked out. And then if we get to apoptosis today, P53 is also going to induce apoptosis. But we'll get to that later. But basically, if a cell gets a lot of DNA damage, the cell generally it depends from cell to cell, but a lot of times when they get a lot of DNA damage, the cell decides to kill itself. It says, I can't probably repair all this damage correctly, and rather it's better for me to kill myself than for the whole organism to get cancer. So that's the kind of decision that occurs based on how much DNA damage, and it varies from the different types of cells. But we'll talk about the mechanism by which P53 induces apoptosis later. Okay, so we're going to be looking at what P53 is doing in cancer. So basically, as I said, in a wild type situation, you get DNA damage. The cell's gonna. So one of the things, of course, P53 is doing is it's not only stopping the cell cycle, it also turns on a lot of genes required for DNA repair. So it's basically saying, okay, stop what you're doing, cell, and just let's repair the damage before you do anything else that could mess things up, like replicate the damage, which would fix it in position, or go through mitosis with damage. So it's going to stop the cell cycle, turn on genes to repair the damage. So that's what this happens here. So this cell gets arrested, repair the damage. As soon as the damage is repaired, there are no more replication forks, which is what's causing the DNA damage signal. P53 gets degraded, and then the cell just keeps going through the cell cycle and starts dividing again. Or there's so much DNA damage that P53 induces apoptosis through a pathway we'll talk about. Cell kills itself. That's the end of the story for that cell. but the organism doesn't have to worry about getting cancer from that cell because it killed itself. Okay, so what happens if a cell doesn't have p53? Then it won't be able to repair the damage, and when you don't repair the damage, that's going to make it more likely to cause cancer. So, but the truth is that, as we'll see, p53 is usually picked up late in the process of making a solid tumor, not early. Okay, it's one of the later events. This would suggest that it's an earlier event, but it is true that if you don't have p53, you're more likely to get mutations, okay, which is cancer promoting. You know, or there's so much damage that they die, but they would have died anyway if p53 was there anyway. But this way, there's no repair pathway. There's no stopping the cell cycle. And once an error gets replicated on the other strand, there's no way for the cell to know that was an error, right? It just gets fixed on the other strand. Okay, and we had talked before the last time about aneuploidy and the role that it has in cancer progression, where the cells no longer act normal. So the cells are starting to, like, you know, they were squamous epithelium, but now they start getting other weird stuff, like now they can move, they couldn't move before, you know, all kinds of things that they shouldn't be doing, degrading extracellular matrix, you know, working in a hypoxic environment. There's so many different impediments to the growth of the cancer, and being able to get new attributes allows them to get past that, and the major way to get the new attributes is become an aneuploid. And almost all cancers that are solid tumors in humans are aneuploid. Okay, so but I didn't tell you how they become aneuploid. Okay, so we're gonna go over that mechanism right now. Okay, so first we're going to talk about this this chromosome breakage fusion bridge cycle, and then we'll talk about the mechanism, because this is part of the mechanism. Okay, so what happens if you have a chromosome and you get a break, okay, in it? So let's say you get the break. So normally you have telomeres at the end, okay? Telomere has a loop, okay? It's protected, but now you get a break. Well, the cell is going to normally say, oh, that's DNA damage signal. It's going to shut down, okay? So it's going to go in senescence, and it's going to be caused by p53. So p53 is going to be like, whoa, broken DNA. We're going to stop the cell cycle until it gets repaired. But there's no repair for a broken end of DNA. Unless there's another end of DNA to connect it to it, there's no repair, right? Because it hasn't actually divided yet. So what we're talking about is a situation where there is no p53. So we're dealing with like a cancer cell, no p53. So there's a DNA damage signal. The cell doesn't care, and it's going to go through S phase. So it's going to make another copy of itself, but it's going to be lacking the telomere, and it's going to have a broken end. So now the cell's like, whoa, I got two broken ends, and I'm going to connect them through non-homologous end joining, which is the repair pathway for broken ends. So it's going to combine them, and it's going to connect them to each other. Okay, but there's a problem. And that problem is that now you have this chromosome with a kinetochore attached to this chromosome with a kinetochore. Suddenly you have a kinetochore on two strands, right? So, I mean, we can see what that's going to look like. And it's not going to be pretty. So normally, chromosomes are being pulled in the opposite direction, right? Sister chromosomes. But now we have a situation chromosomes are attached to each other. So what's going to be the end result of this? It's going to break, but it could break anywhere. It could break here, break here. It doesn't have to break in the middle. I mean, there's no way it can break exactly in the middle. I mean, there is, but it's statistically, super insignificant. It's going to break randomly. So you end up with a broken chromosome, and then over here you got a longer broken chromosome. Okay, so suddenly you've already messed up because they're broken chromosomes, and now you've got a cell with a broken chromosome. Well, what happens to a cell with a broken chromosome? Well, if it doesn't have p53, it's going to replicate itself, and then the cell's going to go, whoa, now I can fix that broken chromosome. I'm going to connect them by non-homologous end joining. And then you've got the same situation. You've got a chromosome that's connected to another chromosome, and they both have kinetochores. And then we're going to end up with the same problem. And it's going to break again. Now it'll break in a different place. And then we end up with the same problem. Chromosome with broken end. So then we go back to here. As you can see, it's a loop, right? That's why it's called the chromosome breakage fusion bridge cycle. Okay? Because it's chromosome breakage. There's fusion. It's going to make a bridge between them. And it's going to break. Right? Chromosome fusion break cycle. So you're like, okay, you know, that's interesting. But how do you get this situation? Where did this come from, right? What, like every single one of those chromosomes broke? That doesn't make any sense, right? How did that happen? How do you end up with this situation where every chromosome is affected? You've got all kinds of weird stuff happening, right? But this happens in almost every tumor, almost every solid tumor. So it must happen like on some regular basis. There must be some mechanism that causes it. Okay, so now we gotta go back to the lecture on telomeres. So we're dealing with, let's say we're dealing with a cancer that's not a stem cell. So it doesn't have telomerase. And you remember that you've got like these chromosomes. They got telomeres. And so this is chromosome 1. Two, 17, you know, there's all kinds of chromosomes, right? So they all have long telomeres, but they're cancerous, so they're dividing too much. So they just keep dividing. What's going to happen to the telomeres when they're not a stem cell and they keep dividing? Because we did go over this in class. Huh?
Eventually, if it keeps dividing, it's not a stem cell, it's going to run out of telomeres, so it's going to start breaking into the DNA, causing DNA damage.
Oh, you're doing the whole thing. I was just asking what's going to happen to the telomeres. Huh? Not chewed up. Shorter. Right. That's right. So you were ahead of me on that one. So they're going to get shorter. So, okay, so like, you know, after this thing, they become shorter. And then after this, they become shorter. So they keep dividing. They're all becoming shorter. But it's not just one chromosome. They're all shorter. Now, what you may or may not remember is that the telomeres form that loop. Okay? So you got the T loop. And then the little one's a D loop. Okay? And this is all telomeres. Okay? The whole thing is telomeres. So the telomere doesn't have a DNA damage signal because it forms a loop. So now, what happens if it gets too short? It's too short to form the loop. So now you end up with an exposed end. Because it's not long enough to form the loop. So that's a DNA damage signal. The cell's going to rest there. And that's the end of the story. Unless p53 is not present, in which case there's no arrest. And so who's going to fuse together? This is not just happening to one chromosome. It's happening to every chromosome. So you could have fusions between 1 and 2. You could have fusions between 3 and 17. You could have fusions between this and this. So now, and then again, all these things have their own kinetochore. So now when they do the chromosome bridge fusion cycle, we're not talking about within chromosome 1. We can have fusion between chromosome 3 and chromosome 5. So this is basically how you're going to get these translocations between different chromosomes, because the ends end up linking to each other and then breaking randomly. Like, you know, so this chromosome maybe broke here and it picked up this one of this dark magenta. And then this one actually did it twice. It picked up a pink and then a green, just based on who it fuses with. Okay? So you end up with this really big mess. And it's just it goes for a few cycles. You just keep scrambling the genome. You're going to lose stuff, overexpress things, and like that. So how would this cell get out of this horrible cycle, which seems pretty bad even for a cancer cell? How would it get out of this cycle? Huh? Well, I mean, that's not going to help the cancer become a cancer. I'm not saying we want to root for cancer, but, you know, from the cancer perspective, I'm not going to root for everybody. Yeah.
Express telomerase?
Yeah, that's right. Express telomerase. So as soon as you express telomerase, then as soon as you have a break, you know, you can just add on telomeres, and then you can, you know, you'll end up stabilizing the whole situation. And you can end up expressing telomerase. So once you express telomerase, then it's sort of a semi-stable state where, you know, sure, you're aneuploidy, but you're a certain type of aneuploidy, and everybody's stabilized like that. So that's the answer. And so generally cancer cells, all the solid tumors have telomerase activated. Okay, so this is the same thing I just talked about, but of course my drawings are much clearer than this is, but you can study this later, but it's the same thing. I just went over it. Okay, now we're going to talk about DNA tumor viruses and some RNA viruses that affect humans. So there's very few RNA viruses that actually cause cancer. They're pretty small populations, like there's this HTLV1 type of retrovirus that causes leukemia in Japan, West Indies, but it's not a big deal generally. I guess hepatitis C makes the liver keep dividing, cause damage, and it can cause liver cancer. But these DNA viruses are much more potent or much more prevalent in making cancers. And like papillomavirus is a major source of uterine cancer. And so for that reason, probably everybody in this audience has the HPV vaccine, or at least all the women do. And I think they're starting to do men too. So while you were a teenager, you got the HPV vaccine because the human papillomavirus, or HPV, is causing almost all cases of uterine cancer. So if you just get the vaccine, and then you don't have to worry about that particular cancer in the future. And there's other ones as well. But again, these are not, like we said before, those retroviruses that are picking up oncogenes. It's pretty much just another animal effect. It doesn't really affect us. But these ones have a different mechanism for causing the cancer, and that is that they're gonna inactivate tumor suppressor genes. Okay, so at least some of them are. So here you have human papillomavirus, HPV. Like I said, you guys are all probably protected against it. That's good. If you're not, you can probably get the shot still. And basically, it makes a protein called E6, and E6 binds and inactivates P53. So it's a tumor. It's going to inactivate one tumor suppressor, right? And so inactivating tumor suppressor promotes cancer to some extent. And then E7 is going to bind and inactivate RB. So you've got a situation where the same virus is inactivating the two major tumor suppressor genes. Like, why would this be helpful for the virus? Why would the virus want to do this? I mean, the virus doesn't care if you have cancer or not. How's it advantageous for the virus? Why would it evolve? Yeah. Well, I mean, right. So the virus is in the cell. Now, if you get more cells, because cells are dividing, then it can replicate during the S phase and replicate like that. So it's just a base for getting more cells in which it can then put out virus particles. So it wants whatever it cells to be in to divide. And also, the E6, of course, is going to prevent P53 from killing itself, because P53 also can induce apoptosis based on having this foreign DNA in there. All right, so a lot of these solid tumors are really slow. Some of them are slower than others. Like I said, RB is not slow. That's why RB is a childhood disease, because the retina is forming in the child, and 2, you just need a single hit of the RB gene and you'll get a cancer. But a lot of these cancers that occur in older people, they have to have multiple hits, and that's why it only happens in older people. Because you're gonna have certain mutations that are gonna cause like hyperplasia or dysplasia where it's overproliferating, like a benign tumor caused by another mutation. Here's a class. So here we're doing colon cancers. This one generally affects older people, so you don't actually have to be screened until you're over 50. And then they put like a colonoscopy thing where they have a little light, and they have a little they actually have a little scissors, and they have a little suction, and they'll go in to the colon, and if they see a polyp growing, they'll just say, I see a polyp, and they'll snip it, and they'll suck it, and then it's like, so it's like 2 things. 1, it's checking if you have cancer, and 2, it's preventive, because these things take so long to develop that, if you can just cut it away, you get rid of, the usual source of that cancer. The cancer's not a problem until it becomes malignant and starts spreading everywhere. So if you can hit these polyps early, then you can get rid of them. So that's why they want adults to go at like every 5 years after they're 50. And it's a pretty good idea because these things, even when they start developing, take a long time. So, every 5 years you can just clear out all of the polyps that are developing, and then, you're less likely to get the cancer later. Okay, so there's certain genes that are going to be causing the early steps in colon cancer. So you have a lot of genes that are mutated, but some of them are going to occur early. And one of the genes that's mutated is APC, but this is not the APC that you saw in mitosis. So that was NFA's promoting complex, and this is adenoma polyposis coli. So it's sort of bad that they have the same acronym, but it's a totally different gene. In fact, this one is part of so this is the Wnt signaling, and this complex that binds beta-catenin phosphorylates it so that becomes a signal for an E3 ubiquitin ligase to degrade it. This adenoma polyposis coli is this APC, okay? So when you knock this out, it's as if you're getting constitutive Wnt signaling, because Wnt signaling disrupts that complex. But if you mutate APC, you disrupted the complex too. And that allows beta-catenin to not be degraded to go in and turn on genes. So just like we saw how extra Wnt signaling causes breast cancer, so extra Wnt signaling through this pathway is going to cause colon cancer. In fact, what it's causing is not cancer per se, it's causing overproliferation. So in this tissue, Wnt signaling is regulating whether the cell divides or not. And as we said before, other tissues are not going to use Wnt signaling. They could care less if you lose Wnt. They're not going to divide more. But in this tissue, the genes that get turned on by Wnt signaling by beta-catenin are going to cause proliferation. So so there's certain families where APC is mutated, both copies. And then what happens is these people get colon cancer super early, and the reason why is because here's a wild type colon, and you got all these folds, but it's pretty smooth. And if you were to have a polyp, you could see it, and that's what I told you where they can just cut it away. But when APC is missing from the beginning, the whole thing turns into a polyp. Well, it's not even a polyp. It's like the first part of this cancer progression is overproliferation. And so this is where every part of the colon overproliferates. And of course, because overproliferation is the first part of cancer progression, these patients have a huge chance of getting more cancer because they have many more cells that are dividing, more chances for mutations. So they're going to get colon cancer super early, right? Because they have the first gene in the pathway. So pretty much APC's mutated initially in colon cancer to start this process. Okay, so these so as I said, the different tumors are going to have different mixtures of oncogenes activated or tumor suppressor genes inactivated, and these things are going to vary between tumors, but it's not all random. Certain tissues will have a high likelihood of certain ones tumor suppressor genes inactivated and oncogenes activated. And different tissues are going to have a different mix. Okay, so like, here's a P16, another tumor suppressor gene, but not RB, right? And this one I have APC mutated. So what this has to do with, or well, let's see if you guys can figure out, what does this have to do with? What's determining this? Like, why are different tissues, why would you mutate, why would they have different mixtures of combinations of genes that would cause them to overproliferate or become cancerous? Yeah. So you're saying a different like kind of mutagenesis or something? Like, like on the skin, you'll have thymidine dimers that, cause a certain type of mutation? I don't think so. Because the different kind of mutations can all mutate genes. But I mean, it could contribute a little bit. Huh? Nobody knows? There's a whole mess of you guys here. What's going on? Oh, yeah, raise your hand. Well, right. So basically, the ability to turn on genes is going to be different, right? And so we had just talked about like the Wnt pathway. And I gave you two examples where the Wnt pathway is going to cause cancer. So breast and the colon. But that's because in those tissues, beta-catenin turns on genes that are required to proliferate. But in other tissues, the beta-catenin can turn on genes that are required to differentiate. So that's actually the opposite. They actually don't divide. So basically, there's the transcription factors and the intracellular pathways that you guys learned about in signaling are going to be tied into different outcomes, based on like who's present. Like what other transcription factors are present for a transcription factor to interact with, right? That's going to change who the targets are. Are some of the targets repressed, or are they accessible, and these kind of things. So you have all these different signals. All the cells are looking for different types of signals to respond in different ways so that they do what they're supposed to be doing in that particular tissue versus the cell next door or the cell in another tissue. They all have different, they're all looking for different readouts to proliferate or differentiate or whatever they're doing. And so you're basically tying into that pathway when you're mutating these, these genes. So, that's why like BRCA1 is important for breast cancer, but not too many other cancers, because it's very important for proliferation in breast cancer, but not in other ones like that. Okay, so we're going to talk now about adult stem cells and their link to cancer, and then we'll talk about cancer stem cells, which are not actually the same thing. So an adult stem cell has an adult stem cell pattern of division. So an adult stem cell pattern of division is you've got a stem cell that produces a stem cell, and it's a non-stem cell. And then the stem cell produces a stem cell and a non-stem cell. And then the stem cell produces a stem cell and a non-stem cell. Okay, so what happened? So we had 1, 2, 3 divisions, but we only ended up with one cell that can keep dividing. Right? So there's no increase in the number of cells that can divide. 3 divisions, it's not 2 to the third power. It's just one cell. So what they're doing is they're producing cells that are not stem cells. Now the non-stem cells might be able to divide like maybe twice or something, but that's it. Okay, so they have a limited amount of division, and this is called replicative, or I think it's called like replicative division. But this is a limited. But the stem cell can just keep dividing forever, but it's not increasing. Okay, we're not getting more stem cells. We still just have one stem cell. So this is an adult stem cell division pattern. In this diagram, that cell division that I showed you okay, where you've got a stem cell producing a stem cell and a non-stem cell. That's right here. So I understand this doesn't look good. That's why I drew my drawing. This thing produces a non-stem cell and a stem cell. Okay, that's this. Stem cell produces stem cell, non-stem cell. So basically, from this pattern, you can get cancer in two ways. So the non-stem cell is going to have a few of these divisions, but they're limited. That's what I drew over here. Okay, and then they're going to differentiate. And that's the normal thing. So the stem cell is just throwing off cells that they'll divide a few times and they're going to differentiate, and that's just to maintain the tissue. And then what you can get a cancer if the stem cells produce two stem cells. Now every cell can keep dividing, right? So instead of a pattern where a stem cell only produces one stem cell, now you get two stem cells. Clearly, that's an exponential increase, right? And then another way you can get a cancer is if these non-stem cells, instead of only dividing a few times, they just keep dividing. Okay, and that's another way to get cancer. So this is a way that you've deregulated the adult stem cell pattern. And these are, and we'll talk about it a little bit more, but usually there's some kind of a niche. And the stem cells, so these cells can do this kind of pattern, but they can also have another one which, is a little bit more, it's not exactly the same pattern, but basically they're dividing here, and while they're in here, they're stem cells, but as soon as the cell leaves, comes a non-stem cell. So it's not really the stereotypical pattern, which you do see in certain situations. It's more like, well, when you're in the niche, you act like a stem cell. But because they keep dividing, they're going to push cells out and they become non-stem cells. So this is actually probably more common. But this is how everybody represents it, right? But this is probably the reality. Like this is what happens in Drosophila germ stem cells. But that's because only one of them can be in the niche. The niche is touching another cell. But in other situations where you got a bigger niche, everybody in the niche is a stem cell, and then they throw them out. It doesn't matter. It's the same pattern, okay? You're still going to end up with basically the same number of stem cells every division, whether it's because there's only a certain number of stem cells fit in here or because they're making this pattern. All right, so that's adult stem cells, and like you said, you can have mutations that are going to cause them to become cancerous, but there's also the cancer stem cell hypothesis. So in that, you've got a situation where, let's get away from all this writing. You've got a situation where you've got an adult stem cell pattern, but it's in the cancer. And so in this case, you got a cancer stem cell that, again, produces a cancer stem cell, a non-cancer stem cell, cancer stem cell, a non-cancer stem cell. So this is based on the fact that if you take a lot of tumor cells, like thousands of them, and you put them into nude mice, you'll get a cancer. But if you just put in like 100 or 10, depending on the tumor, you don't get anything. No cancer. So that means the 10 or the 100 couldn't make a cancer. The 1,000 could. And so the idea was that, in fact, they're not all the same. All the cancer cells are not the same. Only some of them can keep dividing as a cancer. And those are the cancer stem cells. Okay? So these cells can put out cells that can then divide a few times, okay, to make it, and that expands the cancer. But the only ones that are able to continuously divide are the cancer stem cells. So it's like an adult stem cell pattern, but it's not it doesn't have to have come from an adult stem cell. It can just be that this is a cancer stem cell for this cancer. And then another way to look at this is, well, I told you about all that aneuploidy, and these things are unstable as anything, right? It could very well be that these things are so unstable that you're throwing off cells that can't divide, at a certain rate, just because they lost this chromosome or they gained this chromosome. And so, sure, in a population, only some of them can divide, but it's not because there's a particular type of cell. It's because they just got messed up, their genome, like they lost chromosome 3. They can't divide anymore, okay? So the way to distinguish these two hypotheses, and this seems to be working in different cancers, so it's not like one of them is right. Some cancers will have a cancer stem cell population, and some cells won't, okay? The way to distinguish that is if you can find a cell marker, like if you look at the cancers, and often the cancers will have differentiation markers. So like some cancers like teratomas will have they'll actually make hair and muscle and nerves in the cancer, right? I mean, that's an extreme example. But there'll be different markers on the surface. But if you can find one population that if I can sort them away from the other cells, like these yellow cells, almost every one of them can cause cancer. But these ones that are not these yellow cells, they can't form cancer. I can add 10,000 of them. It's not going to help. Okay, no cancer. Whereas here, there is nothing that distinguishes the cells that can form the cancer from the ones that can't. You can't distinguish them. Okay? And so the way you can prove this cancer stem cell hypothesis is to use the fluorescence activated cell sorter that we were talking about before, except now what you're doing is you got live cells and they have certain receptors on their surface, and you're adding antibodies to detect, and the antibodies are fluorescent, to detect like this particular one that looks like a Y shape. Okay, so then now this cell lights up because it's got this Y-shaped receptor, and you got an antibody against that receptor, and then you just use the flow cytometry to say, well, okay, I'm going to segregate all the ones with that Y receptor, and then I'm going to ask, are they able to form cancer at a much higher rate, like just 10 cells versus 10,000 cells? If that's the case, and then the cells that don't have that marker, like that particular receptor on the surface, well, they don't work, then, that would tell me that we're dealing with a separate population that is the cancer stem cell. So that's the gold standard way of showing you have a cancer stem cell, being able to separate the cancer stem cells away from everybody else based on some cell surface marker with an antibody against it, and then show that when you put these in nude mice, and nude mice are just mice that don't have a well-functioning immune system, and so they don't kill the cancer cells that go in, and the cancer can divide if it's cancerous. So these ones you go in, they'll form cancer at low rate, very few cells will cause cancer, and these ones won't. Whereas here, you won't be able to get a marker because they're all just mixture, just an unstable population where there's potentially some kind of epigenetic changes or aneuploidy that's messing things up. All right, and then people are thinking, oh, well, the cancer stem cells might be in a vascular niche. So vascular just means blood, and so I told you about the adult stem cell niche, where when you're in the niche, you're a cancer stem cell, and so people are thinking, well, I don't know whether this is well supported by evidence or not, actually, but the idea was that these cancer stem cells may also be in their own niche, and that niche would be next to blood vessels. So they're gonna throw off cells that are not in the niche. They're gonna divide a few times, but then eventually they'll differentiate. And here they're showing them differentiate into neurons. Okay, so but I don't know how much evidence there is and which cancers this would actually be the case. But clearly they have to have some blood vessels, and I don't know that I go over it in this class, but some cancers, because let's say you're a solid tumor and you're in the middle of it, well, you're going to die because you don't have any oxygen. So you're going to have to turn on genes to overcome that. So there's certain genes that can get turned on that actually make blood vessels, it's called angiogenesis, go into the cancer. And certain genes that control that process get mutated in certain cancers, and those cancers are known for having tons of blood vessels in it. But otherwise, that is an impediment to cancer growth, because in the center, those cells are going to die because they're not getting a blood supply, whereas the ones on the outside will still be okay, but you've got a dead core of the cancer. This is one of the reasons they're talking about putting the cancer stem cells next to the blood vessel. So clearly, the therapeutic target of drug companies is to target the cancer stem cell. So if you just hit most of the cells in the tumor, but you didn't hit the cancer stem cells, they're the ones who are going to remake the tumor, and then they'll just regrow. But if you can target the cancer stem cells in particular, while you didn't get rid of the whole tumor, it can't divide anymore. So that's why a lot of drug companies are trying to go after cancer stem cells. So like, what would be like my idea would be like, well, the cancer stem cell definitely has telomerase active, but that could be true for this whole thing. We don't actually know because otherwise it wouldn't have it turned off. But some molecular marker of being the cancer stem cell is what you want to go after to make sure that the proliferative drive behind the cancer is removed. Okay, so we're going to shift to apoptosis. Hopefully we're able to cover everything in class. We're not going to cover this whole thing. What do we got, like 6 minutes?
Yeah.
I'm drawing too much. I need to stop drawing. I'll stop drawing now.
Yeah, reward the people who come to class.
You want a pop quiz? Is that what you want? Oh, you meant by drawing. Oh, you're saying drawings are good. Drawing is good. Pop quiz is good too. What? All right, we'll do a pop quiz. So, okay, so what question should we do, Snehal? Oh yeah, let me stop recording.